Okay, I will share with you some of the results that we had from the last 5 years, focusing on one example on melanoma brain metastasis, fighting it in 3 dimensions. So a few years ago, in 2015, I heard a talk from Kim Margolin from MD Anderson, where she told us that in post-mortem analysis, 75% are found with occult dormant micrometastatic disease in their brain. This is a really high number. Last year I heard her again, and this time she said it moved up to 90%. I was sure I didn't hear her correctly, and after her talk I went up to her and I said, did you mean 90? 90% of patients, of melanoma patients, have micrometastatic disease in the brain? This is really shocking, because if you think about it, other sites of metastasis like the liver or the lung are more trivial, but when we think about it, why would metastatic cells, meaning those cancer cells that detach from the primary tumor, circulate in the bloodstream, find their way into the brain? This is supposed to be the most privileged, protected site, organ in our body, where the blood-brain barrier is supposed to protect us. So we had an hypothesis that maybe if we think about it, looking at the cells that detach from the primary site, in this case of melanoma, when they detach and start to intravasate into the bloodstream, start circulating, they secrete those cytokines and chemokines, pro-inflammatory growth factors, that while in circulation, they need somebody to respond to them. So it makes more sense when those stromal cells in the liver or in the lung respond to them. But when we look at the residents in the brain, like the astrocytes or the microglia, we see that they can respond as well, and they become activated. This can be seen by GFAP activation of the astrocytes and IBA1 activation of microglia. And this changes their status and actually taking their physiological role of taking care of the homeostasis of the brain. So they become activated, and in response, they secrete other pro-inflammatory cytokines that this time they are preceded by astrogliosis and microgliosis, meaning astrocytes and microglia that start proliferating as they should when there is a traumatic event in the brain or a neurodegenerative disease that starts, or stroke. But we see this neuroinflammation that leads to vascular hyperpermeability, and this is when the blood-brain barrier is compromised, meaning that its integrity is not continuous anymore, but now it becomes leaky, allowing the circulating tumor cells, in this case melanoma, we see the same for breast cancer and for lung cancer, they find their way to extravasate from these leaky vessels and colonize in the brain, create those occult dormant micrometastatic lesions that probably are related, the high numbers are related to the previous talk about the immunotherapy and our immunotherapeutic understanding, that immunotherapy had an amazing revolutionary effect on a certain subpopulation of patients, but in those patients, it allows for longer survival, and this allows the circulating tumor cells to actually reach the brain, colonize there, and then at some point start recruiting their own microenvironment, meaning the microglia, the astrocytes, new blood vessels, and so on. So in order to test this hypothesis, we started by creating models, spontaneous models of melanoma brain metastasis from primary tumors, where we inoculated primary melanoma cells, both from patients' cell lines or from murine cell lines, and following their becoming about 300 cubic millimeter, we excise them and then wait for them to go to other sites. And when we got some brain tumor lesions, we isolated them, re-injected them intradermally to the skin, and then waited for again and again passaging them until they become homed to the brain. And we took those primary and metastatic lesions and compared them by any technological advancement method that you can think of, whole exome sequencing, RNA-seq, RNA single cell sequencing, mutation sequencing. It was mentioned by the two previous speakers about the BRAF V600E mutation. 50% of the melanoma patients bear this mutation. And proteomics and looking at epigenetics, also at DNA methylation. And to make a very long story short, when we find some hits, looking if they are driver mutations, we test them functionally, meaning that if we have, this is oversimplifying it, if we have overexpression in the metastatic lesion of a certain mutation or a certain gene, we knock it down by CRISPR or SH and look if it affects the proliferation, migration, invasion properties, tumor progression, and metastatic ability into the brain, and then by post-mortem analysis looking at the pathology. And if we see that it is indeed a driver, we have two options. One is that we find that there are already drugs that are commercially available for that or in investigational stage. In these cases, we can just repurpose them. It happened to us previously with rosiglitazone and PPR gamma used for diabetes and some other examples. In this case, we can look at BRAF inhibitor, the BRAF-inhib, together with MEK inhibitor that are used in combination, FDA-approved combination for melanoma patients. And what we did here is to combine them together on a single nano platform, meaning that instead of injecting them simultaneously, we make sure that they arrive simultaneously to the tumor tissue, which is not trivial. Each one of these drugs have a completely different half-life. One is 2 minutes, the other is 21 hours. It's very nice when we test them on a plastic dish and leave them for 72 hours to be bathed in with this combination of toxic agents. This is not what occurs in the body. Each one arrives at a different time point, if it arrives at all, and only a promille of the amount of the injected dose obviously arrives to the site and of the primary, let alone of the brain metastatic lesion, where we have another layer of complexity of delivery to the brain. So this allows us to get both of them together simultaneously into the tumor tissue, extravasating due to their nanoscale size from these leaky vessels in the lesions in the brain, and then being cleaved and released simultaneously, exploiting their synergistic activity. In other cases where we don't have available a commercial compound, we can go for siRNA if we want to knock down, or mRNA if we want to express something that is missing in the advanced stage and deliver it by complexing it to a positively charged nanoparticle, where we have a complexation by electrostatic interactions of the negatively charged RNA or DNA. And we can deliver them selectively to the tumor site following IV administration. Then we test them on cells. In this case, you see melanoma cells that are labeled with mCherry, or on the in vivo models in mice. Usually, if we use a human tumor, it will be on SCID mice, immunodeficient mice, or a murine tumor on immunocompetent mice, which is way more relevant, of course, to immunotherapies, and let alone to vaccinations. And in this case, you see the brain metastasis here shown by non-invasive intravital imaging of the red, meaning the mCherry labeled the melanoma cells that arrived spontaneously to the brain, and hopefully this will move to the patients. So first, what we did was to create these models. We had 3 layers of models. The first, a murine model, a BRAF V600E mutated model versus a non-mutated, a human cell line of coming from brain metastasis, and a melanoma brain metastasis sample from a patient from the surgery room. And you can see in all of them that indeed our basic hypothesis was correct. We see the IBA activation of the microglia, GFAP activation of the astrocytes, and CD31 expression, meaning high angiogenic potential, especially, of course, in the case of melanoma brain metastasis in the outer viable rim. But we see all of them, and indeed, as you can see here, by a modified Miles assay and Evans blue extravasation, when it's bound to albumin, meaning that it's 67,000 Dalton, it can extravasate only from the leaky vessels, we see where the blood-brain barrier is compromised, and we allows the Evans blue bound to albumin to be extravasated in those lesions of leaky vessels and compromised blood-brain barrier, meaning those are the melanoma brain metastases. So the first hypothesis, steps of our hypothesis was right, but in this case, we see it on an existing metastatic disease. And we claim that the neuroinflammation precedes the metastatic colonization. So we needed to see the early events. Of course, we cannot get it from patients, but we had some neurosurgeons joining the lab, and they said, okay, let's start from the patients. Take from the surgery room, a chunk of the tumor, implant it into mice, and do everything in reverse. And this worked very nicely. We created a lot of PDX, patient-derived xenograft, but it takes about 9 months to have enough cohorts of different groups with all the controls in order to test. But if we speak about our basic hypothesis in the level of the basic science, to look at the tumor-host interactions, or if we talk about screening drugs or our own drugs or commercially available drugs, 9 months is too long. 9 months, but that time, most patients in this advanced stage are no longer with us, or the tumors that reside in the brain have no similarity to the tumor we excised and worked on for the last 9 months, due to mutations, due to selection, due to the ones that actually survived the surgical excision and the treatment. So this becomes irrelevant, although making a lot of breakthroughs on the way. So we had to think about it a bit differently, and for that, we created a 3D printed platform, meaning that we get a notification from the surgeon that there is an upcoming surgery with a melanoma brain metastatic patient. We get the MRI of the patient, we do the image analysis, we convert the files that we got to STL files that can be read by a 3D printer, and we create hydrogels, polymeric hydrogels, that mimic the extracellular matrix of that tissue, in this case the brain, and we characterize them together with all the cells. In this case, it's not like only the tumor cell layer on 2D rigid plastic that has nothing to do with the actual tissue. In this case, we have the tumor cells, the astrocytes, the endothelial cells, the pericytes, and the microglia, meaning that we have a pentaculture here of 5 different populations of cells in a gel that mimics the plasticity, elasticity, the young modulus, the stiffness of the tissue that mimics the brain. And we create those blood vessels inside the tissue and connect them to a microfluidic system where we can flow the blood from the patients, the PBMCs from the patients, or flow the drugs that we are testing and mimic their diffusion or extravasation from them. So this is how it looks from outside. This is a 3D printed chunk of a brain, but inside what we have is the different cells populations. And you can see it here by the combination of these wonderful 3 PhD students, Lena, who is a chemical engineer, Noah, who is an architect made into a biologist, and Yael, who is an MD student going through her master's studies, where they combined forces to create those blood vessels from a sacrificable polymer, meaning that now we have hollow tubes, tubular structures with a lumen, as you can see here in the Z-stack of the confocal. In green, we have the endothelial cells, in red the cancer cells, in blue the astrocytes, and non-labeled pericytes and microglia that were freshly isolated. And you can see that indeed we can flow by a microfluidic system connecting to a peristaltic pump. Obviously it goes into the incubator and it flows at a rate that we decide that is relevant to the clinic. Moreover, we see that we create the large vessels, meaning around 100 microns in diameters, but they self-assemble and sprout, making their own branches and anastomosis connecting those blood vessels to each other, which is, for me, it's really exciting because I didn't think that we will make them do that naturally. And indeed, the gel allows them to mimic that property in the brain and in the tumors in general. And you can see here that we can even see and detect tumor cells, the melanoma cells, that are stuck in those blood vessels and extravasate from them slowly in those leaky areas. So what we intended to do is now taking these models is to first mimic the surgical part where we created a a smart probe from image-guided surgery, trying to prevent at all the detachment of melanoma cells into the circulation. And the second part that I will talk is the nanovaccine that we developed for trying to prevent or intervene in this case. So first we created, based on FRET property, which is when two molecules of dye or more, when they are in close proximity, they are optically silent, and when they are apart, they will shine and emit photons. In this case, we'll get, meaning that it will be fluorescent at near-infrared wavelength. And what you can see here is that if we inject these probes that are activated by cathepsins that are overexpressed in these melanoma lesions that we found in the first step of our mode of action, we see that if we do a surgery of the primary tissue, the primary melanoma, we get 40% survival when it is under white light, but we double the survival to 80% when we do it with this smart probe injected 1 hour prior to the surgery. And you can see that in those lesions in the brain, we can detect them with the same probe, meaning that this is the reason they succumb to the disease, because it allows, the surgeon basically missed few of the cells that were in the outer region and allowed them to detach, circulate, and extravasate in the brain vessels into the brain to create those lesions. So this was the first step, meaning that we work on the surgical part, but then we know that we have many patients that even though they undergo surgery at an early stage, most of them are coming to the clinic later in stage. So we wanted to make sure that we have something to offer them that will sensitize this population that is not responding to anti-PD-1 for immune suppression blockade, and in this combination we used an anti-OX40, which is an agonist for T-cell stimulation and expansion, and to see if together with a nanovaccine we can get better outcome. And for this, we joined forces with Elena Florindo and João Conhot from University of Lisbon, who are now in sabbatical in my lab, and Anas Comparin, who is now a PI in Turin, in University of Turin. And we created these nanovaccines, nanoparticles at 170 nanometer size, where we entrapped some immune potentiators together with MART1, MHC1, and MHC2 short peptides that are the way pathologists identify melanoma, and we added a mannose targeting moiety to target dendritic cells that have on their surface the CD206, the mannose receptor. This allows that in the site of injection of our vaccine, where we inject it once weekly for 3 weeks, we get the dendritic cells to come to the site of the injection. The vaccine internalizes by receptor-mediated endocytosis, and we get their activation, maturation in the lymph node and processing of the MHC1 and MHC2 peptides. We see that we get recruitment and activation of the T cells, CD4 and CD8 T cells. Here we can see that indeed the dendritic cells got into the lymph nodes selectively following subcutaneous administration. The mannose allows specific and selective internalization into those dendritic cells that overexpress the CD206 mannose receptor. So we moved to in vivo after showing all the in vitro possibilities that indeed those activated T cells can kill the melanoma cells as they home on those MART1 expressing melanoma cells. And indeed we saw that for prevention, when we inject the nanovaccine, then the melanoma cells and then the combination of the immunotherapy, we get inhibition of tumor growth and prolongation of the survival. Then we moved to an intervention setting, and we saw that we get similar results for as an outcome for using just the immunotherapy or just the vaccine. So all the synergistic activity was eliminated in the intervention setting, which was very sad for us because we saw these really promising results from the prophylactic schedule. And we managed to look at all the immune profiling of those tumors that were treated with each one of the possibilities, the vaccine or the immunotherapy. And indeed, we found this MDSC population, myeloid-derived suppressive cells, that are the ones that inhibit the activity of immunotherapies. We found it also in tumor samples from patients that indeed we get MDSC elevation in those patients that do not respond to immunotherapy. So we figured that if we add ibrutinib, a BTK inhibitor that will eliminate this MDSC population, maybe we can improve the outcome. And indeed, to make a very long story short, we inhibited significantly the tumor growth in a specific manner, as can be seen here by ELISpot, interferon gamma expression, and by a tetramer specifically designed for MART1 or for ovalbumin or GP100, different peptides that we used, showing the outcome of prolonged survival. So to conclude, this is a beautiful story from my part, combining clinicians, engineers, biologists, and chemists together, looking at from the bedside into the branch and hopefully back to the bedside. This was done by this wonderful group of people, biologists, MDs, engineers, and chemistry, and showing that the whole is indeed greater than the sum of its parts. Thank you.